Okay, welcome back. So let's start talking about some diversity among the prokaryotes. We're starting with the bacteria, okay? And we are going to talk, we know more about the bacteria, more about them. So here's that original phylogeny where crops, so that we're just seeing the blades of the bacteria. So there is tremendous among prokaryotes bacteria. There are far more prokaryotes than eukaryotes on the planet. Actually, more individuals cells in your gut than the number of humans have ever lived on the planet. Think about that for a minute. There's an incredible range of diversity in the prokaryotes and in the bacteria. Specifically, so in slides, we're going to just kind of go over the main prokaryotes, starting again with the. So this first slide I have up, I don't know if you can see my arrow here. So we've got what we call high temperature clades, the Hadobacteria and the Hyphilic bacteria. Both of these types of bacteria can withstand very high. So the Hadobacteria, thermophiles. So whenever you see that, that means love. So they love high heat. They can withstand extremely high temperatures that are approaching boiling point. There are some members in this clade that can actually, and here's an example of one here, that can actually withstand atomic radiation and even degrade nuclear waste. So they're very tough little critters. Similar in its ability to withstand heat are the hypothermophilic bacteria. You will find these organisms living in extremely hot places such as volcanic vents or hot springs. We even have found some of these guys living in underground oil reserves. So a very tough group. So our next group, and again, we're just going to kind of go through the taxonomy here. And when we get in lab, we'll get a chance to look at some of these in a little more detail under the scope. Not all of them, but some of them. So the next group I want to talk about are the Firmicutes. So here are our Firmicutes on our cladogram, our phylogeny. So these are small, mostly gram-positive bacteria. There are a few gram-negative, mostly gram-positive though. And then there are a few, some, this isn't very common, but there are a few that have no cell wall. So this is a pretty diverse clade in terms of that. Some produce what we call an endospore. No, I don't have a picture. Some produce what we call an endospore. So an endospore is a resting structure that can withstand harsh environmental conditions and regenerate when conditions improve. And when I come back in my next video, after this one, I'll get an image of an endospore for you. Some endospores have proven to be over 1,000 years old and still regenerate, and there are some claims that suggest over 1,000,000 years. So it is a pretty effective tool. So some examples of organisms in Firmicutes, so Bacillus anthractis, so anthrax, the bacteria that causes anthrax is in this clade. Staphylococcus aureus is also in this clade. This is a common skin bacteria that can cause infections in wounds and respiratory and intestinal systems. Sometimes we refer to these as staph infections, so you may have heard of those. Another clade we're going to talk about are the actinobacteria. So our actinobacteria are gram-positive. We have some that are pathogenic, which means they can cause disease. We also have some in this group that are important in producing antibiotics. This group, we tend to get the development of highly branched filaments, which you can actually see in this image right here. So some examples of actinobacteria, Mycobacterium tuberculosis, so the bacteria that can be a cause of tuberculosis is in this group. And as I said, they can, most of the antibiotics many of the antibiotics we have today have actually been derived from actinobacteria clade members. So it's been a really important group for humans for that reason. We've been able to derive a lot of antibiotics from it. So now on to probably my favorite group, the cyanobacteria. So this group is super important because it is the first photosynthesizers. The first photosynthesis in the world came from cyanobacteria. An ancient lineage of cyanobacteria that probably was fairly similar to the cyanobacteria we have today. Sometimes these are also known as the blue-green bacteria. They use chlorophyll as a photosynthetic pigment. And so they're really important because they are a primary producer. And in case you need a refresher, the process of photosynthesis releases oxygen as a byproduct. So the cyanobacteria, ancient lineages of cyanobacteria, were very, very important in helping establish the oxygen in the Earth's early atmosphere and in maintaining it today. So very important. Other things about cyanobacteria that are important is that many are nitrogen fixers. What does that mean? It means they can take atmospheric inorganic nitrogen and convert to an organic form. So they can take nitrogen, say from the atmosphere, so in the atmosphere it would exist as N2, that is not readily available to most organisms, and it puts it into a form that other organisms can use. So that's really, really important. Many cyanobacteria, like the Anabaena you see on this image, form colonies. So what you're looking at is actually a chain of bacteria that are coming together to form a colony. So lots of, sorry, I can't write today for some reason. Lots of colonial species. And these colonies oftentimes have what we call heteromorphic cells. Well, we know the word hetero means different, so difference in morphology. So, and you see this in this picture right here, right? We have some cells look different than the other. So we have these vegetative cells, we have the spore, and we have a cell called a heterocyst, and they all look a little different, and they all play a different role. So the vegetative cell's main role is photosynthesis and to just kind of go about the business of, metabolism. The spore is a resting cell resistant to harsh conditions. So this is an example of very similar to the endospore. The only difference is that the endospore would actually be inside like another layer. that's why it's called the endospore. So that spore is resistant to harsh environmental conditions, and if the environment got really terrible, so the rest of the colony died, the spore would survive, and then when the environment got better, it could regenerate the colony. So it's really important. And then there's this cool cell called the heterocyst. And this heterocyst is specialized to fix nitrogen. So it turns out that the process of photosynthesis and the process of nitrogen fixation don't really play nicely together. So separating, in that some of the byproducts can interfere with the two different types of chemistry. So separating them into two different cells, different compartments, is a really good thing. But they can share their products with each other. So the vegetative cells can share the sugars from photosynthesis, the heterocyst can share the fixed nitrogen. So they work in a cooperative way with one another. So pretty cool little group of organisms are the cyanobacteria. All right, let's talk about the spirochetes. These are gram-negative bacteria with a spiral shape. So these are really distinctive looking. So all possess this axial filament and a bundle of flagellae beneath their peptidoglycan layer that allows for movement. So if we were to kind of cross-section them, we would see this cluster of internal flagella. And so what this cluster of internal flagella allow them to do is kind of move with this twisting motion. So they kind of have a really kind of interesting mode of motility and internal anatomy. So these ones are really nice and distinctive in that way. Many are parasites, and many cause diseases, including syphilis and Lyme disease. So not always the nicest of organisms, but definitely really cool little organisms. All right, chlamydius. I bet you can guess what this causes. Okay, so this organism is sort of unique among prokaryotes because they have this kind of complex life cycle where we have two forms of cells. So complex life cycle. There's what we call reticulate bodies and elementary bodies. So let's take a look here. So here we have an image. So here is an example. So again, we have kind of the two cell forms. We have the elementary bodies that get taken up into, in this case, it's taken up into a eukaryotic cell. And then once it's in there, it develops into a more thin-walled reticulate body, which has the purpose of growing and dividing. And then these reticulate bodies will reorganize themselves into elementary bodies, which then get liberated with the rupture of the host cell. So they just kind of have this more complex life cycle that involves two forms, is the basic take-home. So these types of bacteria cause a number of infections in eyes and lungs. This can be a cause of pneumonia, and of course also can cause STDs. So watch out for those. Okay, so the final clade of bacteria I want to talk about are the proteobacteria. It would help if I had my marker cap up. So these are the largest and most, or is the largest and most diverse clade. Mostly G negative, so mostly gram negative, using light energy to metabolize. Sulfur, but we also have nitrogen fixers. Really, it's just such a big clade. There's a lot of variation in lifestyles here. So some of the best known pathogens are members of this clade, and some of the best known bacteria in general. So E. coli is one of the most well-studied organisms on Earth. This is in this clade. Some very famous pathogens, such as the pathogen causing the bubonic plague, or cholera. So cholera, which is caused by a bacteria called Vibrio cholerae, that's in this clade. Salmonella is in this clade, which of course is a cause of food poisoning. So we have a lot of different types of organisms in this very large clade, more than we can really get into in this class, but you definitely need to know the clade. Okay, so now we're on to domain Archaea. Okay, so domain Archaea. So this is much less known, so less well known. And one of the reasons for that is many of them are what we'd call extremophiles. So an extremo, that E shouldn't be in there, sorry for my bad spelling. An extremophile lives in a habitat that is not habitable by other species. And we do have some bacteria that, you might call an extremophile, like our high temperature clades, but it's really common in the Archaea to live in a really unhospitable environment, so that just makes them harder to study. So we will talk about a few different groups here, and I'm kind of going to talk about them in terms of their sort of habitat. Now, I should mention that the group that is genetically determined to be the closest known living relative of the eukaryotes is this group, Lokiarchaeota. So I would kind of know that name. In terms of the rest of the Archaeans, we're more going to talk about them in terms of the habitat we find them in as opposed to their clade name. Okay, so... oh, sorry, I'm ahead of myself. Let me go back. Alright, so let's talk about them. I thought I had another image for Archaea, but I guess I don't. So let's talk about some of these different Archaean groups based on their habitat. So we have what we call our thermophiles. So our thermophiles live in extremely high heat, like more than 131 degrees Fahrenheit, like extremely high heat. We have what we call our acidophiles that can live in extremely low pH, such as less than 0.9. That would be more acidic than stomach acid. We have our methanogens. So these would be anaerobic, so not using oxygen. So these are anaerobes that generate methane as part of their metabolism. So when they reduce CO2. So they generate methane when we'll just say when they produce energy. So as part of their metabolism. And they actually create 80 to 90% of all the methane in the environment, and many of them are found in the guts of mammals and are responsible for producing the gas that is passed from a mammalian digestive system. So you can blame your archaeans next time you're in an elevator and somebody points to you. And then finally we have the halophiles. So these are all our salt lovers. So they live in very salty water. Often, and so we have a picture of this back here. So here are some, the water is actually pink here due to the carotenoid pigments that these archaeans tend to have. And this would be an environment that other species would die in because they would dry out due to osmosis, right? The water would leave their cells and go to the saltier place, but the halophiles have evolutionary adaptations so that does not happen to them. So again, our archaeans, we have our thermophiles, our acetophiles, our methanogens, and our halophiles, and we're going to kind of categorize these guys based on those environmental conditions. Okay, so I'm going to end this lecture, and I'm going to pick up and have our final, I know, there's a lot. Our final chapter 25 lecture is going to be all about some ecological and nutritional adaptations we see in the prokaryotes. So stay tuned.